So we're going to go back to some basics again. And I'm going to pick up on some things that Dr. Dietrich presented, especially in sequencing and FISH, and we'll go into some deeper detail on those technologies and techniques. I realize that many of you do these techniques in your laboratories, so you're quite comfortable with many of these and very familiar with them. But as you also probably recognize, some of the things that are the very basics and fundamentals, we sort of forget over time, or we can't quite remember how to say it. And some of the techniques are going to be unfamiliar to you. If you only do one type of analysis in your lab, you may not do the other. So we're going to go into some detail about the basics of these techniques. When you're starting out looking for mutations, there's a couple of questions that you need to ask yourself before you even talk about what technology or technique you're going to use. And one of those fundamental questions is, what material are you starting with? Because clearly when you're starting with a paraffin tissue block, it's very different than starting with a blood sample, for example, and your technologies are going to be more limited based on your starting material, particularly for paraffin embedded samples. You've heard over the course of both days all of us say negative things about paraffin embedded samples. I mean, we all have said it. Now, don't get us wrong. We love our paraffin tissues. I love my tissue. I love my slides. However, they do limit us in certain ways because of those fixation problems that have been talked about over and over again. The second question you need to ask yourself is, what mutation are you detecting? What type of mutation do you want to detect? Because that, again, will inform the type of assay that you'll perform and looking for the specific mutation you're looking at. Very common types of mutations that we look at in oncology all the time include point mutations. You've heard that over and over again. You've heard KRAS and BRAF and EGFR, all of which have point mutations in them. We also are often looking for today translocations. The ALK-EML4 gene is a translocation that's very commonly done today in clinical practice. But there are many more coming. And I'll tell you, as we learn more and more about tumors, it's surprising how many solid tumors have translocations that were never really recognized before. In the last 3 or 4 or 5 years or so, in my field of head and neck pathology, we have found translocations in very high numbers in adenoid cystic carcinoma. Who knew that? That's a surprise that 60% of adenoid cystic carcinomas have a consistent translocation. In mucoepidermoid carcinoma, 60 to 70% have a translocation that's very consistent. Clear cell carcinoma of the salivary gland. And the new tumor, mammary analog secretory carcinoma, which was discovered because of its translocation. So we're learning that translocations are much more common in solid tumors than we ever thought before, and have very important therapeutic roles. So many of these translocations pair up oncogenes that can be targeted with therapies, with something that's driving its expression and activation. So we actually like translocations not just for diagnostics, but actually for therapeutics as well. Of course, you're very familiar with amplification from the story of HER2, which we're not going to really touch on today, but we will talk about detection of amplification mutations. And then deletion mutations. Now, deletion mutations have a long and complicated past, with the years of looking for loss of heterozygosity as a surrogate marker for looking at tumor suppressor genes. But I'll also just call your attention that one of the common EGFR mutations is a deletion mutation, not a point mutation, and that's an important role as well for mutation testing. And then lastly, most of us are familiar with the story of KRAS and BRAF in colon cancer treatment, but we sometimes forget that one of the most important tests we do in colon cancer is detecting people who have hereditary disease. And microsatellite instability is a form of mutated DNA. It's a form of mutated DNA that's a consequence of having a primary mutation elsewhere, but it's certainly something else we like to detect at the tumor level as well. So we'll talk about all of these technologies in a bit of detail. Point mutations are associated with both hereditary disease and with oncogenes. And the oncogene mutation panels that we usually associate with this are somatic mutations in the tumor itself. Now, mostly in anatomic pathology, this is what we're looking for, and we're not really looking for the primary hereditary disease. So we're going to focus on point mutation detection in tumors, and in particular in paraffin-embedded samples. Oncogenes are commonly referred to as dominant genes because only one copy of the gene needs to be mutated in order for tumorigenesis to occur. These are activating mutations. They're frequently either point mutations or amplifications, or very frequently translocations involve oncogenes that are driven inappropriately by something else. Oncogenes, again, occur in one copy of the gene. So whenever you think about detecting these mutations, recognize that there is one wild type and one mutant in oncogenes. So you will always be looking at something that is heterozygous in a sample. And that's important when you start thinking about the technologies you'll utilize for detecting these oncogene mutations. Most oncogene mutations, actually probably all oncogene mutations, are activating mutations. The mutation occurs in the gene, which leads oftentimes to overexpression of the protein product. And this is interesting, but not diagnostically very useful, because overexpression is not a reliable correlate to the primary mutation. So we can't use that overexpression necessarily as an indicator that the gene is mutated. Overexpression can be caused for a variety of reasons. But overexpression due to oncogene activation often leads to things we associate with tumors: cellular proliferation, angiogenesis, apoptosis, things that are tumorigenic in their nature. And this is one of the reasons why many pathways that are involved in tumorigenesis involve oncogenes that lead to this type of growth and proliferation of tumor cells. So how do we detect point mutations? Well, we detect them with a lot of different technologies, and you've heard about a few of those from Dr. Dietrich, and you've also heard about some of the pitfalls and problems in those technologies as well. If it's a reasonably consistent, small, and reproducible region, it's a lot different than the scenario of a larger region, such as exons that need to be analyzed, because if you have a very targeted area like a BRAF mutation, you can apply something like pyrosequencing, which only sequences a very short distance from the primer site, as opposed to a gene that might have mutations spanning across multiple exons, where you really need to look at the entire exon in order to detect these random mutations or more random mutations. And in those cases, you're looking at something like Sanger sequencing or even next generation sequencing to take it to the next step. I will say, since Dr. Dietrich's no longer here, that I am not as optimistic as he is that next generation sequencing will replace what we do today. I think it's worth learning the basics and fundamentals, because I think you'll be using them for quite a number of years. I remember not too long ago when everybody said, oh, microarrays and expression profiling, that's going to replace everything we do. We're never going to do RT-PCR again. We're never going to do quantitative PCR. Forget it. No point. We have arrays. Have they? Anyone doing arrays clinically? No. Why? Why don't we? It's expensive. It's too big. We don't need that much information. The information is information overload. And we know that there are problems with the starting material that you've heard about over and over again. So I think we will see the same sort of trends. We will see applications of next generation sequencing, and certainly the targeted approaches are going to be very powerful and strong. But the whole genome analysis, decades away for practical purposes. So we're going to spend time talking about Sanger sequencing. You have seen this pathway over and over again, and I want you to ingrain it upon your brain. It needs to be burned into your brain. This is a pathway, EGFR, but all the receptor tyrosine kinases signal through this pathway, that is oncogenic. It's in its nature, it is involved in most oncogene pathways that we talk about in tumorigenesis today. So the RAS, RAF, MEK, MAP kinase pathway is just one that pathologists need to know about. It's so critical in our understanding of tumorigenic pathways today. And we're going to talk about the mechanisms to analyze many of the different components in this, especially the ones that are oncogenic. So KRAS has been mentioned over and over again, codons 12 and 13. If you do this type of analysis, you know that GGT, GGC, backwards, forwards, every which way. Actually, that green box got shifted a little bit. It's actually GGT, GGC. It goes over just a little bit too far there. This is common. This is what we look at all the time for KRAS mutation analysis. And the way we can do that is highly variable. Again, depending on the starting material and depending on what information and what output you need at the end. There are detection methods that don't require actual detection of the mutation. Those screening techniques like SSCP that we used to do all the time. Why did we used to do that instead of sequencing? because sequencing was expensive and not everybody had a sequencer. And you didn't want to put every sample into the sequencer when it took hours and hours and you only had a limited number of samples. Now our sequencers are fast and efficient. You can put many samples on at once. It's cheap, it's easy. So that technology really has replaced some of those screening techniques we used to use. We used to pre-screen and then only do a selected few. And now we really just sequence. It's just so much more straightforward. Allele specific PCR is another method that can still be used very, very efficiently, especially if it's a very specific mutation and we know what it is and it's very consistent. For example, BRAF is a great example of a simple allele specific PCR that one could do. Then, of course, the full sequencing technologies. At least, I say full sequencing, but at least they're getting the sequence, and that's Sanger sequencing, which really is full sequencing. Single base extension, or the snapshot technology, which is also using the same technology as sequencing. Pyrosequencing, which you heard about earlier, and next generation sequencing, which you've also heard about. So let's start off with the pre-screening, or those methods that we used to use for detecting mutations without actually saying it was a mutation. And those really mostly rely on conformational changes. So a mutation at the genome level will make a shift or a funny shape in the gene or in the sequence, and that will mean that it will migrate at a different rate or a different speed or a different way than the normal. So really we would stick this mutant in and we would stick the normal in, and it would migrate in an altered fashion, either with heteroduplex formations or with the actual conformational shape change. This didn't tell us what mutation was there. It just told us there was something different. And you can imagine what polymorphisms did for this, of course, because a polymorphism would show up exactly like a mutation. So there's no way to tell what the mutant is in this technology. If you got a positive when you did this, you would then go and sequence the ones that looked positive, and you would find out if the mutant was there or not. Allele-specific PCR is a simple technique which requires you to build your primer to incorporate the mutant site. So if the mutant site here for BRAF is right here, the black, red, black, then you would include that in your PCR primer. And one primer would have the mutant, and one primer would be normal. And when you amplified both, preferably in separate tubes, but possibly multiplexed if you design them correctly, then you would get two different products if you have a mutant and a normal present. So you would have one product amplified from the normal primer, and you'd have one product amplified from the mutant primer, and you'd see two different products and know that one mutant and one wild type were both present. And that's also a very straightforward, simple approach. Using capillary electrophoresis, this is a very, very strong way to look for a mutation that you know and that's very specific. The advantage of allele-specific PCR is actually quite significant when it comes to sensitivity, because allele-specific PCR is actually more sensitive than Sanger sequencing. You don't need a 20 to 30% tumor ratio for allele-specific PCR. You still probably need a 5% tumor ratio, so it's not perfect. It certainly is not as sensitive as you would want for something like minimal residual disease or something where you're looking for rare tumor cells. But again, the gold standard for all mutation detection comes back to a fundamental technique, and that is Sanger sequencing. Gene sequencing, or Sanger sequencing, is also called dideoxy sequencing. And I'm going to spend some time talking about this technique at the very fundamental basic level again, because I don't think it's going away, and it actually underlies our understanding and ability to perform all the advanced sequencing technology as well. I also want to talk about translocations in particular, because translocations in specifics are going to become a very important technology and application that we use in tumor-specific analyses, more and more and more. In terms of translocations, we also see these in rare hereditary diseases, but we don't really use that analysis in anatomic pathology. What we really look for in tumors is the somatic mutation, or the translocations, where we know both partner genes or we may not know both partner genes. So in terms of the translocation analysis, we expect two genes to be partners in the translocation. Gene 1, the DNA, and gene 2, the DNA, become swapped or become converted into one. And so we expect these translocations generally to occur with breakpoints in the introns, so that the exons become put next to each other from two separate genes. This is somewhat of a problem, because if you know much about introns and exons, you recognize that introns are very large and exons are very small. So going back to the fundamentals of what kind of DNA we can get out of these samples, you recognize that using a PCR across these very large areas is going to be nearly impossible from paraffin-embedded samples. So if you look at the gene 1 and gene 2 next to each other at the DNA level, you will have a very large area of DNA between here, and amplification will be nearly impossible across that area. Remember that the average size of fragments from paraffin-embedded samples will be 200 to 400 base pairs in size. You'll never get a PCR product across that, which is why we're forced, if we're looking at translocations at the nucleic acid level, to use RNA rather than DNA so that you can amplify across that. Now, once the RNA is processed, the introns become spliced out, and 2 exons from 2 separate genes become put next to each other. PCR is possible across that by converting it to cDNA and then getting a product and doing polymerase chain reaction after that. So this is why we can use mRNA analysis or RT-PCR analysis to do the translocation analysis. Now, I want to come back to this concept of EGFR in this pathway, just to remind you that one of the things we've been talking about over and over again is the ALK translocation. The ALK translocation is important in lung cancer, and it also signals through the same pathway. ALK is a receptor tyrosine kinase signaling through exactly the same pathway as the EGFR. There's much more to this story. This story is not complete until you recognize that there are also now ROS translocations that are known, RET translocations that are known, and also other things like MET amplification, which is a resistance mutation that's known in this pathway as well. So you can see that translocations are going to become a very important thing that we're looking at. We've talked about ALK and EML4, and we've talked about the fact that it's a translocation that occurs in lung cancer. And you also have heard recently from Dr. Dietrich especially that it's actually not a true translocation. It's an interchromosomal rearrangement, which is incredibly annoying. I mean, it is very inconvenient that it's a rearrangement when it comes to the analysis part of that, and he mentioned that problem as well. The ALK gene is fairly consistent and has a translocation in a fairly consistent spot, but the EML4 breakpoint is much more diverse and variable within the gene, which leads to some problems in designing an assay, because you can imagine that if you wanted to design an RT-PCR assay for this, how many different primer sets would you need? A lot. Unless they're really close together and really small exons, you're going to need multiple different primer sets in order to detect this at the RT-PCR level. I think somebody asked that question yesterday, and I wanted to point that out, that this is the answer why RT-PCR is very challenging if you're looking at this particular example. Now, most of them, 62%, will be these two, so you could pick up most of them by doing a targeted analysis. However, it is much, much easier to think about doing this at the FISH level and doing it from fluorescent in situ hybridization, much easier approach. So in situ hybridization is a pretty powerful technique. It allows us to look at gene fusions in 2 different ways. In one system, we can do fusion probes, where there's 1 probe on each partner, and in the abnormal scenario, the 2 probes will come together. On the other scenario, we can do break-apart probes, where the 2 probes flank the consistent breakpoint in 1 partner gene, and in the abnormal scenario, they come apart, and that's why they're called the break-apart. So you do actually need to know critically which probe system you're using before you sit down at a fluorescent microscope to analyze, because it's very critical to know whether you're expecting them to come together or you're expecting them to come apart in the scenario that's here. Fusion for translocation example here. So this is the fusion gene system, where you have 1 probe on 1 partner gene and 1 probe on the other partner gene. And in the abnormal scenario, they come together, and green plus red often looks like yellow, and you'll see that fusion of the 2 probes in the abnormal. Break-apart probe is exactly the opposite. They're normally next to each other. They flank the area that you expect to break in the translocation. And in the abnormal scenario, they're going to come apart or break apart and look like they're separated at a distance. Amplification can also be assessed by FISH, and one of the most powerful technologies for looking at amplifications is by doing in situ hybridization. And now, in fact, it doesn't even need to be fluorescent. You've seen pictures of silver in situ hybridization, which can be very useful for amplification mutations as well. Amplification mutations by FISH are very simple to assess as well. A very simple probe technology where one probe sits on the gene of interest and the other probe sits somewhere else, like at the centromere or even on the other chromosomal arm. And we're looking simply for an increase in the number of the copy signal. And usually, in most amplifications, it's very clear how it's amplified or not amplified. It's either abundantly amplified or not amplified. There's not that much in between, although there are cases that we need to count and actually give a quantitative assessment. I also want to spend a moment talking about deletion mutations. Deletion mutations have traditionally been looked at in tumors that we're looking at tumor suppressor genes. Tumor suppressor genes are different from oncogenes because in tumor suppressor genes, both copies are mutated in order for tumorigenesis to occur. These mutations are often point mutations on one copy, but on the other copy, they are often deletion mutations, which is why typically and traditionally loss of heterozygosity was used as a surrogate marker for tumor suppressor gene mutations. We now also know that methylation is another aspect that can inactivate a tumor suppressor gene. But importantly, they're called recessive genes because both copies of the tumor suppressor gene need to be mutated in order for tumorigenesis to occur. So point mutation on one, deletion mutation on the other, and you can expect for tumor suppressor gene pathways to be inactivated by this mechanism. There are multiple different ways to look for loss of genetic material, and one of those is loss of heterozygosity. But another powerful technique is in situ hybridization, just as we've looked at already with fish or even with silver or chromogenic in situ hybridization. And then lastly, some technologies now are out there that are array-based, and the array CGH is a great example of a technology that one can use for looking at global amplifications and deletions across the entire genome. You've seen this picture before when we talked about capillary electrophoresis and gel electrophoresis and the comparison of the two. And we talked about how you can look at the size of the two products, and we also talked about how you can look at the relative quantity of the two products. And this relative quantity is one really powerful tool for looking at loss of genetic material, particularly with loss of heterozygosity. So when we're doing loss of heterozygosity analysis, we actually are not looking for the gene of interest. We're actually usually not looking for a tumor suppressor gene. Rather, we're looking for a short tandem repeat that's nearby that will be used as a surrogate marker for looking at the loss of genetic material. So if you imagine a short tandem repeat that's in a sequence of gene that's otherwise normal, one of these two alleles is going to be larger than the other, and it's going to be larger by the number of repeat difference. So if we design our primers to sit on either side of that short tandem repeat, one PCR product will be long and the other PCR product will be short. It doesn't really matter what the sequence of that PCR product is. It doesn't matter at all. All that matters is that we have one long one and one short one. And when we analyze that with capillary electrophoresis, what we will see if there's loss of genetic material is that one of the peaks will be very small in the tumor as compared to the normal sample. If you did this by gel, you could also see it, but it's actually very hard to detect those subtle differences. Capillary electrophoresis is much more powerful for detecting loss of genetic material in this manner. So you can look for loss of genetic material with capillary electrophoresis just like this. Now remember, you can lose either allele, so it doesn't matter which allele is lost. One will be high and one will be low, and that gives you the analysis of the detection difference. This is what it looks like in capillary electrophoresis in terms of a real electropherogram with two peaks and with one where genetic material has been lost. Now I show you this because most of us don't do capillary electrophoresis for loss of heterozygosity anymore. It's not a very common technology. But this is the way that the EGFR mutation deletion can be detected, because we'd expect to see two different length PCR products with the deleted allele versus the normal allele. So very exactly the same technology, just a slightly different application. Now in situ hybridization can also be used for deletion mutations. And in deletion mutations, what we would expect is for one of the signals to be gone or absent, and to see two reds and one green, for example, when one of the chromosomal arms has been lost or the gene of interest has been lost as well. Now I'm going to end by talking a bit about microsatellite instability, which again is another mutation or sort of mutation that we detect in tumor samples. It's associated with Lynch syndrome tumors, which most commonly will be colon cancer, but also now we're being asked to analyze endometrial carcinomas as well, and maybe you are too. Now, we talked a few minutes ago about short tandem repeats. Short tandem repeats can be used for LOH analysis, but they can also be used for microsatellite instability analysis. A short tandem repeat is a microsatellite. It's the same word, same name, same genome area. These are short sequences of DNA that vary from about 2 to 7 base pairs in length. Most common ones that we analyze in microsatellite instability are mononucleotide repeats. Some techniques also analyze dinucleotide repeats. It's a short sequence of DNA that's repeated for a variable number of times. So you can have an allele with 4 repeats, or you can have an allele with 6 or 8 or 10. It's highly variable across the population. When you're looking at short tandem repeats, mononucleotides tend to be the ones we like to look at in colon cancer. It's important to recognize what the normal pattern will look like. The normal pattern of an electropherogram for a mononucleotide repeat looks like this. It has a peak in the middle, which is high, and it has stutter down to the right and stutter down to the left. And that's an important normal pattern to recognize. Dinucleotide repeats usually have stutter down to the left only, and usually it's 2 or maybe 3 little peaks to the left. And tetranucleotide repeats have one large peak and then one stutter band peak right below it at about 4 nucleotides to the left of that. Now, when you look at microsatellite instability electropherograms, it's really simple to interpret them if you know what the normal looks like. So normal's up on top, tumors at the bottom, and what you see in microsatellite instability is an altered migration pattern. You see new PCR products from the microsatellite instability as reflected in this electropherogram. So these abnormal peaks here, abnormal peaks here, and abnormal peaks here are all examples of microsatellite instability. And this is another form of mutation detection that we do today. And I'm sorry I eliminated the slides on sequencing because I was thinking that Dr. Dietrich was going to cover those. So I think that if you want to see sequencing slides, I could probably pull them out and we could slip them in another session. It's only about a 2-minute thing, so we can decide that later. Thank you.